This is Dr. Ming Tan. He's a postdoctoral researcher from Southeast Asia, where he studied regional Orthoptera for over a decade. This is kind of a stunning fact here. As a taxonomist, he described more than 100 species new to science. Whoa. Um, his PhD topic focused on pollination by Orthoptera, and more recently is focused on researching the bioacoustics of Orthoptera. And prior to this was a research fellow at the Paris Museum, where he worked on the bioacoustics and communication behaviors of crickets. So I shall hand you over.
Good day, everyone. Thank you for coming to attend this talk. My name is Ming, and I'm a postdoctorate researcher from Singapore. I have been studying grasshoppers, crickets, and katydids for more than 10 years, and I have worked on the taxonomy, ecology, and behavior at different times of my research career. Today, I will share with you a pet project of mine, looking at the discovery of katydids that produce ultrasonic calling songs. I started looking into the ultrasonic bioacoustics of katydids since 2018, very much thanks to my visit to Fernando at the University of Lincoln. He studied the biophysics of katydids and demonstrated to me how katydids can produce extraordinary high-frequency calls, even though they might be small and fragile-looking. In fact, his study showed that more than 70% of katydids communicate with ultrasonic calls, but his study focused mostly on katydids from the neotropic regions. On the other hand, nearly nothing is known about the bioacoustics of orthopterans from the hyperdiverse and highly threatened tropical forests in Southeast Asia. Southeast Asia, as shown on the left, has been considered one of the noisiest places on Earth. This knowledge gap inspired me to build an acoustic library for Southeast Asia katydids. This will include recording the katydids in captivity so that the sound will be of high quality and very low noise level. This will also mean that I will collect the katydids and eventually properly preserve and identify them before vouchering the specimens in museum collections. This is essential as a way to cross-reference the calling songs and the physical specimens. Lastly, collecting the specimens sometimes also leads to the discovery of species new to science. Secondly, beyond just stamp collection, I'm also interested to look at more hypothesis-driven objectives. For example, I would also like to use the acoustic data to test species boundaries and validate existing species defined using only morphology. This is part of an integrative taxonomy approach. To record the ultrasonic calls, I will need recorders that are sensitive to high frequency, as well as something that I can bring around during overseas fieldwork. One of the equipment that I use is the Echometer Touch Pro, as shown here. I will also bring along a temperature logger because ambient temperature can be an important predictor for sound frequency and other acoustic parameters. Recording is usually done in a makeshift, sound attenuated room inside my own apartment. I will try to record each katydid in isolation over numerous nights so that I have ample recording for each individual. And because many katydid species are usually rare and low in abundance, it's not easy to get a big sample size for each species. So having multiple recordings of the same individual in a way is also crucial. One of the interesting activities associated with this project is to keep these katydids as pets. So in my own house, I have an area dedicated for taking care of these katydids. Most katydids will feed on cat or dog food pellets, but some of the carnivorous katydid requires live food, so it also means that I will have to rear fruit flies and feed them. After the songs are recorded, the next step is to describe as well as to analyze them. I usually use Raven light to visualize the oscillogram and spectrogram, as well as to make measurements involving the time domain. This includes, for example, the syllabus duration. I also use packages from R, such as C-Wave, which is very useful for accurately extracting the frequency data and visualizing the frequency power spectrum. I will show you examples of these output later. So in the remaining part of the talk, I will show to you some of the findings and publications from this project so far. First up, I looked into the bioacoustics of Plugidini, a tribe of katydids from Southeast Asia. I recorded the calling songs of 6 out of 13 known species at the time of study. This study has great significance, personal significance to me, because it is the first time I would know the calling songs of the first new species that I described in 2011. As you can see from the habitats of the 6 species, these katydids are very similar looking morphologically, with some small differences in their genitalia parts that you wouldn't be able to notice from just these photos. The calling songs of these 6 species are also similar in that they are made up of rapid decay pulses, which means that each pulses lasts almost instantaneously. But different species produce very species-specific temporal pattern that you can see on the right. When we look at the frequency power spectra, we can first observe that the entire call spectrum of all 6 species are ultrasonic. This means that their calls are completely inaudible to us. But different species also have different peak frequency, ranging between 35 to 60 kilohertz. Beyond just describing their calling songs, I also look at the stridulatory files on the forewings to examine how sound-producing anatomy can influence the differences in the calling song that we have recorded. The stridulatory file, as you can see on the right side of the presentation, is a modified base that is usually responsible for the sound production and the parameters that we can observe, among other structures on the forewings. I made measurements such as the number of teeth, as well as the intertooth distance, which is the distance between consecutive teeth, and the length of each tooth. And based on this graph, one can see that different species differ drastically in terms of their stridulatory anatomy. In another study, I described the calling songs of 24 katydid species from around Southeast Asia and compiled them into a single paper. Most of the calling songs have not been known before, and here is the list. This study is particularly close to my heart as well, because a lot of the data was collected during a difficult time during the COVID-19 pandemic, where I had to shift my study into my own apartment during the lockdowns. From this histogram, we can see that more than 80% of the species that have been recorded from Southeast Asia produce calls with dominant frequency in the ultrasonic spectrum. These include 3 species producing extreme ultrasonic calls, that is, calling songs with frequency peaking at 40 kilohertz and above. I will next showcase a few interesting species, but you can have a look at this open access paper published in the Journal of Orthoptera Research. The call structure of the katydids that have been recorded in this study is highly diversified. It ranged from very simple house-like call of like this species, as well as what you have seen in the previous studies among the Plugidini. But the pulses of these species are usually more spaced apart, and they are also of a lower frequency. So the hypothesis is that this type of call is a way for katydids to avoid detection by bats. Quite a bit of work has been done on the predator-prey interaction between bats and this subfamily of katydids from the Neotropics. In this study, we also produced 3D images of the stridulatory anatomy, including the stridulatory file on both the left and the right tegmina, as well as the mirror area. As you can see, katydids have a unique asymmetrical stridulatory file on both wings, which is quite different from those in the crickets. In crickets, both the right and the left tegmina are symmetrical and they have very similar morphology. Here is a Cornocephalus species which produce a rather complex call compared to the previous species. You can see that there are two types of pulses, either occurring in single pulse or in doublets. If you remember the previous species, the frequency spectrum of the call has a very narrow range of around 30 kHz. This results in a very pure tonal call. But in these katydids, the frequency range of this katydid is pretty broad. We call it broad spectrum, and sometimes the call is considered noisy. Up next is a Phaneroptera species, Phaneroptera brevis, which is quite widespread in Southeast Asia. Interestingly, you can see that there are two separate parts in the stridulatory file, a shorter one where the teeth are more closely packed and a longer one where the teeth are larger and more broadly spaced. What we notice is that the cause is also made out of two pulses. Our hypothesis is that each pulse is produced when the scraper hits each different part of the stridulatory file. We can confirm this if we have a high-speed camera to record the calls as well as looking at how the wings move. The next species of interest is this Argentini fine-headed katydid. This species, endemic to Singapore, has a unique call in that it has a harmonics structure. So if you focus your attention on the power spectrum, you will see that there are actually multiple peaks of frequency, usually in the multiplications of 2. So in this particular species, the dominant frequency, which is also the fundamental frequency, is around 12 kHz. The next harmonics is actually at around 24 kHz, albeit at a lower power. And you can also observe a third harmonic at around 36 kHz at an even lower power. This is interesting because the existence of harmonics is very common among crickets, but very rare among katydids. The interesting frequency spectrum that we have recorded can probably be attributed to the stridulatory files or other sound-producing organs, such as the mirror membrane. So looking into the biophysics will allow us to better understand how this structure can contribute or result in a very different calling structures that we observe in different species. Among the 24 species that were recorded in this particular paper, this species has the highest peak frequency of around 54 kHz. We have not been able to identify this katydid to either the genus or the species, and it is likely that it remains an undescribed species awaiting to be properly named and described. The taxonomy of this group of katydid is also quite complex. So once again, this highlights the importance of collecting the physical specimens. Without the specimen, there will be no way we can identify it correctly, as well as to verify the identification in the future if there is a need to. You may also notice in my first slide that there is actually a plus sign after the acronym D.U.S.K. This is because beyond looking at katydids, I also have a great interest in the bioacoustics of crickets. They are often found together in the same place when I was looking for the katydids. Very similar to the katydids from Southeast Asia, the calling songs of most species of crickets from this region has never been documented. So for the third and last publication that I will share with you, I'll focus on the bioacoustics of scaly crickets. In this study, I recorded and described the calling songs of 7 species, including 1 new species, all of them from a small island state of Singapore where I come from. Once again, as expected, even though the crickets may not look very different from one another, their calling songs are species-specific, both in the temporal structure as well as the frequency spectrum. This is very important so that species found coexist together are able to communicate correctly with only their own species and not members of another species. From the power spectra, you can also see that the kids produce highly tonal call, so they actually sound very melodious. Because multiple species can actually occur in syntopy, that is in the same location, it allows me to look at how species partition their calling song parameters such that they can coexist and that the songs do not interfere with one another and consequently affect the ability of each species to communicate with conspecific. Here I'm showing you 3 species occurring in a single small patch of mangrove in Singapore. Ornithius lupus, which is the new species, Ornithius rufonigrus, as well as Ornithius tampanis. You can see that the syllabus duration, syllabus interval, Akim's duration, as well as the peak frequency of Ornithius lupus and tampanis are clearly partitioned and often not overlapping at all. Both species can be found on the same kind of mangrove tree species and calling concurrently. That their core parameters are not overlapping allow the coexistence of both species. This is also in line with the prediction of the acoustic niche partitioning hypothesis. However, Ornithius rufonigrus seems to falsify this hypothesis because the core parameters actually overlap with both other species. But the reason is probably because Ornithius rufonigrus is found further away from both species in the back mangrove rather than the actual mangrove. So even though they are found on the same site, Ornithius rufonigrus technically do not coexist with Ornithius lupus and Ornithius tampanis. Because of the time limit, this is as much as I can share, but in the past 5 years, I have already published more on the bioacoustics of katydids and crickets from Southeast Asia. Some of these studies are also part of my postdoctoral stint at the Paris Natural History Museum. If you're interested in them, you are more than welcome to contact me. So with this, I would like to thank you for taking time to listen to my talk.
Thank you so much for that. I just got sucked into that whole presentation. It's the first time I've seen it. Usually I try and watch these beforehand, but I'm like, oh, it's really interesting. I like the way that you had to sort of take during lockdown. You had to sort of bring your laboratory home, in effect. Yes, hope you've got an understanding household. Anyway, so if there are any questions, which they're starting to come in, please pop them in the Q&A and I'll get started with the first one because they're already coming in. So what other features are you using to differentiate species? Are you using DNA analysis or physical characteristics?
So I'm primarily looking at the morphology. So this usually requires a close examination of the abdominal apex of the specimen, and very often I also need to carry out genitalia dissection because the genitals are usually the most diagnostic character to differentiate and identify species. Um, I have a bit of experience with using DNA barcoding to identify them, but this is far and few for among the species that I'm dealing with. So for identification, mostly via morphology.
Cool. How willing are most orthopterians to sing in captivity? Do you usually get recordings within 24 hours of captivity?
It depends on the species. Some species are really brave and outspoken. So they might actually start calling on my way back in the small containers. They are more than willing to start calling. But there are also many shy species that didn't even call throughout their stay in the captivity, and then they usually die after a few days. So I think it really depends on the species. Um, yeah.
Oh, and were you in lockdown? Were you recording at home with
Yeah.
trying to get all the other ones to be quiet while you're trying to record one specific one?
So I will have to isolate the individual that I'm actually recording so that the calls of other species are not interfering with the specimens of interest. So yeah, so yeah, that's about, yeah. So some I'm also not too sure if different species might interact with other species. So whenever I do a recording, I will make sure that that individual is isolated. There's no external noise, be it other crickets or other human anthropogenic sound. Yeah.
Yeah. That's a tough job. um, can you explain a bit more how katydids make their sound?
Okay, so Katydids as well as the crickets, they have two, um, they have the four wings, they produce sound using their four wings, as compared to grasshoppers, which use their hind legs. So in crickets and katydids, um, usually there are two stridulatory files which I have shown in the presentation. So when the two wings starts to vibrate or rub against one another, um, these two these two files will be rubbed against one another with as well as with other parts of the forewings. And in that process, sound is being produced because the teeth uh, on the files actually cause production of sound waves. But there are also other structures around the forewings or within the forewings that helps to amplify the sound or modulate the frequency of the sound. So sometimes the ultrasonic spectrum is only possible because of certain modification in the forewings that allows amplification or production of ultrasonic sound. There are also
But-
other species that actually has other mechanism that amplify or maximize the sound. So some of the pronoterms are enlarged, so it functions as amplifier. Yeah.
So does that mean you get um, sounds which enhance, say, a harmonic, but sort of reduce the fundamental?
Yes, there are some crickets that have very unique harmonics. Usually the harmonics have the dominant frequency has the dominant frequency and the fundamental frequency being the same. But there are certain groups of crickets that produce dominant frequency in the second or even the third harmonics. So that
Wow.
is pretty unique, and I think um, there are active research going on trying to look at why these particular group of crickets produce such unique harmonics that are different from uh, typical cricket.
Oh, cool. How long does it take for the captive specimen to acclimatize? Use acclimatize. How do the acoustic characteristics of the captive compared to the free-ranging ones? Are they very similar, or is it completely different? I suppose.
So for acclimatization, Like I've mentioned earlier, I think it really depends on the species, but I will always try to make them more comfortable by adding some plants inside the cage. Um, because we come from the tropical area and the forests are really humid as compared to out in the city or out in the house. Usually I'll cover them up with a wet cloth over the cage to provide that the a more quiet, a darker environment as well as a more humid environment for them. So it works for some species, it doesn't work for other species. Um, for the second part of the question, which is, sorry, can you repeat the second part of the question?
Yeah, it's disappeared off my list.
Uh,
Oh, how do the acoustic characteristics of the captives compare to the
ah,
French ones, to the wild
um,
ones?
so I did do recordings out in the field as well, setting up the Ascend Mini out in the field over a period of time and looking at the acoustic diversity in the natural community. Typically, the we can be pretty sure that the frequency doesn't really change a lot, so long the environments are relatively similar because frequency are dominantly affected by not just the, in terms of environmental factors, it's usually affected by temperature and sometimes the humidity. So if we are able to have very similar environment when we do our recording in the lab, the differences shouldn't be too big. Um, in terms of the temporal parameters, usually parameters within the schemes or within a single unit of calling songs shouldn't differ drastically, such that we misidentify the species. But having said that, I have a very small sample size of the true diversity of the species in this region. So there might be many species that have very similar calls that we cannot differentiate as well. Um, so generally, I think there is not too big of a difference such that we have issue trying to identify them.
Excellent. This is quite an interesting question because I was looking at the acoustic niche that you were mentioning earlier on, and some people adhere to it, some people don't. Some people, some insects do and don't. Did you notice different species changing their singing rates to avoid masking when put in close proximity?
Um, I'm not sure about this because I haven't actually looked specifically into this question. So yeah, I'm not too sure I'll be able to give a correct or good answer.
That's fine.
Yeah.
um, is a database of Southeast Asian Orthopterian sounds recordings accessible online?
Yes, so I have been sporadically uploading whatever data that I've collected and published onto the Orthoptera species file. So for people who works with orthopterans, I think they will be familiar with this database. And recently, this database just migrated to a new platform. So I think it might take a while before I'll be able to upload more up-to-date data. But I also understand that very recently, Canto is also one of the database that allows people to upload autocurrent sound. So um, depends. I might want to focus on one of the database so that it's much easier for people to get all the data at one single place.
Yeah. No, and Xenocanto is a wonderful resource, I think, for I'm a bat guy, and they started doing bats now, so it's like it's even better. So it's great. And we're going to run out of time, but I've got one last question. Can you describe your equipment for recording katydids?
Okay, so I uses the Echo Meter Pro Touch. So it's the one that is attached to the mobile phone. Um, it is so usually I would place the recorder, i.e. the phone with the recorder right beside an insect cage, um, less than a meter, because once the distance is too much, is too great, the sound quality might be affected. And then inside a sound attenuated rooms, dark and sound attenuated room, I'll leave it to record usually overnight. So most of the species from our region produce sound at night. And then hopefully the next day I will look back at the data and look back at the phone, and hopefully I'll get some recordings. Of course, there will be some other noise that I will need to filter out. For the crickets, using the Echo Meter Touch Pro will not be very ideal because that equipment is more suited for recording ultrasonic calls. So for cricket that produce low frequencies, so something like 5 to 10 kilohertz, it also means that this recorder is not very useful at recording these low frequencies. So in this species, for this case, I would actually use the normal sound meter. So uh, that usually allows me to record a frequency up to 45 kilohertz, which is more than good enough for most crickets, such that we can not only see the dominant frequency, but we can also see the harmonics, which sometimes can reach around near ultrasonic range.
Thank you so much. Well, thank you. There are more questions in the Q&A, so please feel free to and please keep asking questions as well, because I'm sure you're going to be quite happy to answer them. Really interesting talk once again. Thank you very much. I'm sure there's going to be lots of those little symbols going up. There you go. Look at that, right on cue.
Thank you.
Thank you very much. And